Assalamu alaikum. Dear students, today we will discuss about cell and cell division. The nature is so diverse and advanced that we merely consider about the origin and evolution of living things. Have you ever thought that what is the basic component of all living things? In fact, cell is the basic unit of all living things. So we can say that cell is a basic unit of structure and function of all living things, excluding viruses. On the basis of origin and complexity, we divide cell into two types, prokaryotic cells and eukaryotic cells. Prokaryotic cells are basically those cells which do not have nuclear membrane around their genetic material, and they lack all membrane-bounded organelles. For example, bacteria and blue-green algae. While eukaryotic cells are those cells which have their genetic material membrane-bound by a nuclear membrane, and they contain all membrane-bounded organelles. For example, all unicellular and multicellular organisms. Now let's discuss about the cell theory. The credit for the cell theory goes to Robert Hooke, a 17th century British scientist. He used a simple microscope to study common objects. And besides common object, he also studied and examined a thin slice of cork, which is basically a part of outer bark of cork oak. Later in 1824, a British scientist, Henry Dutroy, he confirmed the cork's observation as cells. So far, we have discussed about the discovery and definition of cell. Now let's discuss about the cell theory. According to diversity, the cells has been divided into two types, animal cells and plant cells. Basically, it took a century and a half to progress from the Robert Hooke's initial observation of cork to the modern cell theory. The credit for the cell theories goes to two German scientists, M.J. Schilden and Theodor Schwann in 1939. According to modern cell theory, there are three specific points. Number one, all living things are made up of one or more cells. Number two, cells have got a basic living things which have got chemical reactions, and all chemical reactions of the life goes within them. Number three, all cells arise from the pre-existing cells. You can see here different multicellular plants and animals, and all these cells and all these plant bodies have made up of small cells. If we see under electron microscope, we see different components of cell. New cells are formed by the division of mother cell. Actually, different parts themselves are not alive. Basically, life is an emergent property of the specific arrangement of cell parts and their integrated activities. The basic components of cells are three: plasma membrane, cytoplasm, and nucleus. So first of all, let's discuss about plasma membrane. A cell is surrounded by an outermost surrounding wall, which is flexible and fatty material, which is called plasma membrane. Inside plant cells, there is an outer wall which is called cell wall and which is secreted by the cytoplasm, and it is made up of cellulose. Plasma membrane basically allows the useful material to get in and the toxic material to exit out of the cell. The plasma membrane, besides performing the barrier activities, also receives and generates signals which help in coordination and communication between the cells. The thickness of plasma membrane is 0.1 micron meter. Structurally, the cell membrane or plasma membrane is a very complex structure, as you can see in the model. We say that the plasma membrane is basically composed of lipid bilayer sandwiched between two protein layers. So this is what we call a unit membrane or plasma membrane. Now let's discuss about the fluid mosaic model. The fluid mosaic model is the most accepted model, and it explains that the plasma membrane consists of lipid bilayer and two tiers of phospholipid molecules, with hydrophilic ends oriented outwards while hydrophobic tails oriented inwards. And facing the hydrophilic heads are arranged protein and glucose molecules which together form glycoprotein. Plasma membrane is basically a kind of membrane which is selectively permeable in nature. It allows the transport of molecule with active and passive transport, while the bulk transportation is done by phagocytosis and endocytosis. The next layer outer to the plasma membrane is the cell wall, which is present only in plant cells. The cell wall is basically composed of three layers: secondary wall, primary wall, and middle lamella. Primary wall is basically composed of cellulose, pectin, and lignin molecules, while secondary layer is composed of inorganic salts, waxes, and cutin, while the middle lamella holds them together, meaning the primary lamella and the secondary wall, they are composed together by middle lamella. And it is composed of basically pectin material like calcium pectate. The basic purpose of cell wall is to give a definite shape and a rigid structure to cell structure, meaning it is only present in cell of the plants, which is giving them a hard and a specific and rigid structure. Cellulose molecules are arranged in a crisscross arrangement, and the function of cell wall is to give a rigid and definite shape to plant cells. The next thing, which is next to the plasma membrane, is cytoplasm. Cytoplasm and nucleus together form the bulk of the cell, which is called protoplasm. The plasma membrane and nucleus together form a very small portion of the cell, while the major bulk portion of the cell is formed by the cytoplasm. Cytoplasm is a highly organized semi-fluid substance which acts as a ground substance for the chemical reactions of cell. About 70% of the cell cytoplasm is composed of water molecule, while 15 to 20% of the cytoplasm molecule consists of different types of protein molecule, which range nearly 10,000 different types, while 10 billion different types of protein molecules occur in an average cell. Now let's discuss about the component of a cell. Cytoplasm contains different cell organelles like endoplasmic reticulum, ribosomes, mitochondria, Golgi apparatus, lysosomes, plastids, and centriole. So first of all, let's discuss about endoplasmic reticulum. Endoplasmic reticulum is a tubular network of tubules extending throughout the cytoplasm. And the tubes of this extending network is similar to plasma membrane. Endoplasmic reticulum was first discovered by Keith R. Potter in 1945. The function of these endoplasmic reticulum is that it provides the rigid structure and support to all the cell organelles within the cytoplasm. And the second function is that it also helps to transport different materials within the cells. There are two morphological forms of endoplasmic reticulum. Rough endoplasmic reticulum, this one, with attached ribosomes, which basically provides the site for protein synthesis, while smooth endoplasmic reticulum, this one, which is lacking ribosomes. Ribosomes are tiny granular structure which are found freely dispersed in cytoplasm like this, or they are attached to rough endoplasmic reticulum. Ribosomes are composed of protein and RNA. The next component of cell is mitochondria. These are tiny spherical rod-like structure which are found in every part of cells. Their structure is very complex, as you can see here. They consist of two membrane structure. The outer membrane is smooth, while the inner membrane has got infolding inside the chamber, and these infoldings are called cristae. Mitochondria is basically used for the production of energy, which is used to build up and to fuel the different activities of cell parts. Basically, it provides the necessary energy for all the cell organelles, and that's why we call it a powerhouse of the cell. The next component of cell is Golgi apparatus or Golgi complex. It is a set of smooth membranes stacked together into flattened sacs, and it contains molecules like protein, carbohydrate, glycoprotein, and some specific enzymes. In some part of the cell, it fuses with the plasma membrane as well. The function of this Golgi apparatus or Golgi complex is to store, modify, and pack the cell secretory products. The next component of the cell is lysosome. These lysosomes are tiny spherical structure found in cytoplasm. They were first isolated by Didier in 1949. These tiny structures called lysosomes contain powerful digestive enzymes, and this helps in autophagy, phagocytosis, and extracellular digestion. The next important part which is present inside the cell is called plastid. But this specific part called plastid is only present in plant cells. Its function is to trap sun's energy and store the food in the form of carbohydrates and other products which are used by all the animals and living beings on earth. There are three types of plastids: chloroplast, leucoplast, and chromoplast. Chloroplasts are basically the green light absorbing pigments containing chlorophyll. And these chlorophyll basically absorbs green light in the form of complex molecule called carbohydrates. The structure of these chlorophyll is very, very complex. This is composed of two membranes. The outer membrane is smooth, while the inner membrane contains thylakoid vesicles, which form the stacked membranes. And these thylakoid vesicles are present in stroma and constitute granum. And the function of these granum is to trap sun's energy in the form of molecules called carbohydrates. And as we have discussed earlier, that these carbohydrates are basically used as a source of energy by all the living things on the earth. The next important part of the plastids is chromoplast. Basically, chromoplasts are the colored plastid. They trap green, orange, and red energy of the light. They are mostly found in all the colored part of the plants. For example, the petals of the flower and all the colored parts of the plant. And the last of the most important of the plastids is leucoplasts, which are also called white plastids. Now, why do we call them white plastids? Because they have got no color trapping pigments. Rather, they contain starch storing pigments, which are mostly found in underground parts of the plant and stored food material. For example, the potato contains billions and billions of the leucoplasts inside them, which contains starch granules. The next important component of cell is centriole. This one. The centriole is further consists of 9 microtubules, and each microtubule further consists of 3 tubules. The function of this centriole is basically the formation of spindle, meaning this spindle formation occurs in cell division. These centrioles are present only in some microorganisms and lower plants. Though all the components of cell are very, very important, but the most of these important one is the nucleus. So nucleus is so important. Why? Because it controls all the activities of the cell and the organelles. Nucleus is further divided into different components. This one. As you can see here, that nucleus can be further divided into different parts. Number 1, nuclear membrane, nucleolus, nucleoplasm, and chromosome. First of all, let's discuss about the nuclear membrane. Nuclear membrane is nearly similar to plasma membrane, but the only difference is that as case in the plasma membrane, there was only one lipid bilayer, while in case of nuclear membrane, there are 2 lipid bilayers with a space in between them. Basically, the nuclear membrane consists of 2 membranes, inner membrane and the outer membrane. And these membranes are perforated by each other with nuclear pores. And the function of these nuclear pores is the transmission or the transport of material from the nucleus plasm to the cytosol. So this is the way how the transportation between the nucleus and the cytoplasm is being regulated by the nuclear pores. And there are dozens of nuclear pores around the nuclear membrane. The next important part of the nucleus is nucleolus, which is spherical in structure. It helps in the synthesis of ribosomes and synthesizes ribosomal RNA. The fluid-like matrix inside the nucleus is called nucleoplasm, which contains the specific enzymes for the DNA replication and for the synthesis of RNA. The thread-like network which is spreading throughout the nucleus is called chromosomes. These chromosomes are composed of a complex which is called chromatin, which is formed by the complex of protein and DNA. These chromosomes are basically called the genetic characters, or they contain genetic character which help in the inheritance of any trait. These chromosomes basically provide the genetic material for the production of new individual and maintains the characters from one generation to the next generation. According to cell theory, new cells arise from pre-existing cells. So there are 2 main types of cell divisions, mitosis cell division and meiosis cell division. So now let's discuss first about mitosis cell division. There are plenty of different types of changes which are occurring in nucleus and cytoplasm in this mitosis cell division, but for our convenience we have divided this cell division into 4 phases, naming prophase, metaphase, anaphase and telophase. But before I proceed about prophase, let's discuss about the preparative phase, which is called the interphase. In this phase, the chromatin threadwards, which I told you about the chromosome, they start to condense, and this is called the interphase. Prophase has been further subdivided into 2 phases, early prophase and late prophase. In early prophase, what happens actually, that chromosomes, they condense themselves and they duplicate themselves, and this is the way how the thin threads which were originally present according to a network inside the nucleus, now they have been condensed into thick chromosomes which have been now duplicated. In late prophase, these thick duplicated paired chromosomes now arrange themselves and give their appearance more and more by condensation. In the end of late prophase, the nuclear envelope disappears and centrioles start forming spindle fibers. Now, next phase is the metaphase. In metaphase, the nuclear envelope disappears. Spindle fibers are formed and the chromosomes are attached by their kinetochores to spindle fibers, and they are arranged on equatorial plate. Now, the next phase is the anaphase. In anaphase, the chromatids are spread from each other and they move to the opposite poles. And the last phase is the telophase. In telophase, the nuclear membrane again reappears around the chromosome, and this is how the cytokinesis also occurs. And one single cell, which we call mother cell, has now been divided into 2 daughter cells. That's why we call mitosis cell division a kind of cell division which always maintain diploid number of chromosomes. So by this, we come to an end, and goodbye.